Hi everyone. Okay, so I apologize for being so late. It's been a busy day for me, but this short little announcement here is to help you to understand how to inoculate your culture tubes, okay? For the two exercises that you'll be doing this week. You want to go ahead and get these inoculated and get the bacteria growing because it may take up to 2 days, and this is not even part of the experiment, right? This is you're going to do the antibiotic discs experiment, and you're going to need living cultures for that. So what you're doing here is you're taking your little vial of E. coli and Streptococcus and you're going to reconstitute them. If you look there at the bottom, there's a little dry pellet. That's the bacteria. And so you're going to reconstitute them with the tryptic soy broth. So if you have been keeping your tryptic soy broth in the refrigerator, make sure that you take 2 tubes out. You're going to need 2 tubes, and let them come to room temperature. Takes about 15-20 minutes, but let them come to room temperature. And then you can use that broth to reconstitute your dried bacteria. And then you're going to use the reconstituted bacteria and you're going to inoculate it back into the tryptic soy broth. And then you're going to grow your bacterial culture in that tube, in the tryptic soy broth tube. I'll explain that, how to do that in a minute. Now, I'm going by the McGraw-Hill instructions. And if you go to the McGraw-Hill and you open that up, if you scroll down, there's some introductory information, some good background information, some little illustrations. That's good information for you to read over. That'll help you in lecture as well. But if you scroll down, then you will come to 2 videos. The first video is the one that I'm focusing on right now, but the first video is the lady there is explaining to you how to handle the bacteria, how to reconstitute the dry bacteria, and how to inoculate your soy broth, excuse me, your soy broth tube. Okay. Now, her bacteria is in plastic tubes. So your kit comes with this little tiny, cute little alcohol lamp. You can use a candle if you want. But it comes with this already full of alcohol. You can see my little wick. My little wick there is dark, so I've already used it. Um, but she uses plastic vials. Okay, so she can't sterilize the lip of the vial. But these little vials that you and I have received are glass. So once you get them open, you can and you should sterilize the lip of the vial as well as the lip, like she does, as well as the lip of the soy broth tube. Okay, you want to do that every time you take the lid, the top off. You want to sterilize the lip of the tube, of the glass tube, because every time you take that top off, there you can pick up some microorganisms just from the air. So anytime you take take the tube off, take the top off, you want to sterilize the lip in the flame. So now our little tubes here have this little aluminum cap on them. At the top, you can't see it here, but you'll be able to see it on yours. There's a little arrow, and that little arrow points to a little uh, a little slit in the aluminum cap where you can get your fingernail easily get your fingernail up under there. You'll see it. It's open. You can get your fingernail up under there, and you can lift up the top of this, um, and and then you just pull it off. The whole little aluminum cap comes off. Once you flip that little lid up, then just grab onto it. You can use a pair of tweezers or forceps or a pair of pliers if you, but get your kid to do it. Doesn't matter. Um, but you flip that little lid up, and the whole aluminum cap comes off, and then underneath you will see a rubber stopper. Now you can take the rubber. You want to take the rubber stopper off, and that will expose the opening to the little to the little bottle. And then you can sterilize the lip of the little bottle. Then you use your little disposable pipette, little disposable pipette. Mine's been soaking in alcohol. These are disposable. They're only single use. And you get a milliliter or so of the soy broth from the tube. You know I'm doing this with one hand. You inoculate or you reconstitute the little pellet. Takes about 20 or 30 seconds, not very long for that thing to dissolve. Sterilize the lip of your little bottle. Go back in with that same. You don't want to sit this down on the table, because you can't sterilize the end of this little plastic pipette. But you go back in with the same pipette. Go back in with the same pipette into this little bottle. Retrieve all of the contents of this little bottle. Sterilize the lip of your tube and inoculate the rest of your broth with the reconstituted bacteria. Sterilize the lip of your tube, put the cap back on. You're going to put the cap back on loosely. Don't tighten it down. Just put it back on loosely so that nothing from the air, no fungal cells or anything, can get into the tube. And then you're going to take your two tubes. I have mine right here, my two, because I did E. coli in one and Staphylococcus in the other. And I'm going to incubate those. Now, I know you don't have an incubator at home, and that's fine. Neither do I. I have, surprisingly, I have a lot of stuff here, microscopes and all sorts of stuff, but I don't have an incubator. I'm going to take my tubes and on top of my refrigerator, it's usually pretty warm. It's close to the ceiling. It gets warm up there. The refrigerator, the motor from the refrigerator heats things up back in that corner. So I'm just going to put mine on top of the refrigerator. Um, but what you want to do is you want to put them in a fairly warm, not hot, but fairly warm and dark place. Remember, that's what bacteria like. They like moisture, they get that from the broth. They like dark and they like warmth. Okay, so that's where you want to grow your bacterial cultures. Um, and and that's the way you handle it, okay? So keep everything aseptic, keep everything as sterile and as clean as possible. By the way, your kit comes with these gloves, and these are like for my little nephew. I can't, I could not get that sucker on. These are like at best medium. They're pretty small. My hands take an extra large glove, so I could not wear those. Very important though that you maintain asepsis as much as possible. So clean your surface. Um, I use my wet wipes for that. Clean it off, dry it. Make sure you wash your hands really well, and then you can handle everything. And by the way, you may have gloves at home that you can use, and that's fine. But I don't, and these do not fit. So What do you do? Okay, so that's that, and I now you're going to grow your culture, incubate your cultures. The literature says for 48 hours. Put it in there, check it after 24 hours. The little solution should no longer be clear. It should be turbid. It should be cloudy. And if it's cloudy, then you know you have bacteria growing in there. Is a good sign. That's what you want. And then you're going to use those cultures to do the antibiotic discs and those kinds of things. So you don't need your auger plates at the moment. Okay. So that's that. Get them in there. Get them incubating. Check them after 24 hours or so. Definitely after 48. If you don't have cultures after 48 hours, and I don't know what we're going to do because, but they'll grow. They'll be fine. This has worked for me before, so not a problem. But make sure you incubate them in a preferably dark corner of your house, in a warm spot. I'm putting mine on top of the refrigerator. Okay, so that's that. Let me know if you have any questions or concerns. I'll be back tomorrow. I think tomorrow with a more descriptive video. And also, I'm going to show you how to do the antibiotic disc experiment as you're waiting for your cultures to bloom. All right, that's that. Let me know. 804 Use my campus email. You can use the Halo inbox right here. Talk to you later.